guys, I am Vijay, and in this video we shall learn about the factors that affect enzyme activity. One of the factors that affect enzyme activity is the substrate concentration. When the substrate concentration is low, then the rate of the reaction is very slow. But as the substrate concentration increases, the rate of the reaction also increases and the products are formed at a much faster rate. Now, how do we know this? Well, we have run experiments and we've seen this happen. So let me explain the experiment to you briefly. What we do is we take reaction mixtures that contain different concentration of the substrate, like 0 millimolar, 50 millimolar, 100 millimolar, and so on. And to these reaction tubes, we add a fixed quantity of the enzyme, and then we allow the enzyme catalyzed reaction to occur. And after a short period of time, we determine the quantity of the product that is formed in each of these tubes. And from this, we can calculate the rate at which the product was formed. For example, if about 6 micromolar of the product is formed in 1 liter of the reaction mixture after about 10 seconds, then the rate of the reaction is 6 by 10, that is 0.6 micromole per liter per second. This is the rate of the reaction, also known as the initial velocity. And what we need is the initial velocity, because the rate of the reaction will not be the same throughout the reaction period. Initially, when we still have the specified quantity of the substrate, the velocity will be high. But as substrates get used up, the rate will slow down and eventually become zero. Which is why we measure the rate as soon as the enzyme and the substrate are mixed, when all the catalytic activity is still going on. During this period, the products are forming actively and we can determine the rate at which this is happening. Now we can plot these values on a graph with the substrate concentration on the x-axis and the rate of the reaction on the y-axis. So let us plot some values here, some values that we might obtain for a typical enzyme. When the substrate concentration is about 5 millimolar, the rate of the reaction is about 33 micromolar per minute. And when the substrate concentration increases to about 10 millimolar, you see that the rate of the reaction has also increased. And now the products are forming at 50 micromolar per minute, which means that the rate of the reaction increases along with the substrate concentration. And it increases sort of linearly. So the initial part of the graph is like a straight line. Now you're probably wondering why this happens. Why does the rate of the reaction increase along with the substrate concentration? Let me explain this to you using one of my favorite tools, an analogy. So let's take a tennis ball and let's mark a red spot on it. Now you place an army ant on this ball. How soon do you think the ant will find the red spot? It might take a while, right? If you have 2 or 3 ants, it would still take some time for them to crawl on top of the red spot. Now imagine this ball is swarming with ants. How soon do you think an ant will find the red spot? Pretty much as soon as they arrive, right? Okay, let's go back to the enzymes. Let's take the enzyme hexokinase as an example. This is an enzyme that uses glucose as a substrate, and it adds a phosphate group to the glucose molecule to form glucose 6-phosphate in the first step of glycolysis. Now the actual size of a glucose molecule is about 10 angstrom. But what do you think the size of hexokinase is? Yes, it is at least 15 to 20 times greater than the glucose molecule. What is shown here is an actual representation of their relative sizes. Imagine this and you can intuitively understand why the rate of reaction increases along with the substrate concentration. Going back to our analogy, the army ant there is like the glucose molecule and the tennis ball is the enzyme. And what do you think the red spot that we drew on it is? Yes, it is the active site. So when there are very few glucose molecules, the rate at which they find the active site is slower, and therefore the rate of the reaction is also slow. But when there are lots of glucose molecules, they find the active site a lot faster, and therefore the rate of the reaction increases. But will the rate of the reaction increase indefinitely as the substrate concentration increases? Not really, right? After the whole ball is covered in ants, it doesn't matter how many more ants join the party. The rate at which one of it is crawling on top of the red spot will be the same. Similarly, after all the enzyme active sites are filled, it doesn't matter how many more substrate molecules are added to the reaction mixture. The rate at which the reaction will take place will be the same. Now let's see what this looks like in the graph. So initially, as the substrate concentration increases, the rate of the reaction increases and you get this linear graph. But after it reaches a particular substrate concentration, the graph flattens off and it forms a plateau. So as you can see, there is a maximum velocity that a reaction can reach. And any further increase in substrate concentration will not result in an increase in the velocity of the reaction. And this velocity is called Vmax. And this graph here represents how the substrate concentration affects enzyme activity. Enzymes have varying affinity for their substrates. Some substrates have a very strong interaction at the active site and have a high affinity for their enzymes, whereas some others have a weak interaction at the active site and have low affinity. Can you imagine how the graph changes in each of these scenarios? Let's take a look. So let's add to this the graph of two other enzymes, one with a greater affinity for its substrate and another with a lower affinity. Let's assume all three have the same Vmax value, which of course is very rare. Vmax value changes from enzyme to enzyme. But for simplicity's sake, so that we are comparing apples to apples, let's assume all three have the same Vmax value. So enzyme 1 is already drawn here. Let's draw enzyme 2, which has a greater affinity for its substrate. And its graph would look more like this. As you can see, it attains greater velocities at lower substrate concentrations. Whereas enzyme 3, which has a lower affinity for its substrate, would have a graph that looks more like this. And as you can see, it attains the same velocities only at a greater substrate concentration. Now we can quantify this. If we measure the substrate concentration at half the maximum velocity. Half the maximum velocity for enzyme 2 would be this point, and if we drop a perpendicular from this point to the x-axis, we will get the substrate concentration at half the maximum velocity. Now this value is a constant. It is called the Michaelis constant and is represented as Km. The Km value is lowest for enzyme 2, which has the greatest affinity for its substrate. Whereas enzyme 1 has a higher Km value and enzyme 3 with the lowest affinity for its substrate has the greatest Km value. So Km value is an inverse measure of enzyme substrate affinity. There are some substances that can hinder the activity of an enzyme. These are called inhibitors. There are many different types of inhibitors, but one of the type is a competitive inhibitor. An example for competitive inhibition is the inhibition of enzyme succinate dehydrogenase by a substance known as malonate. Succinate dehydrogenase is an enzyme of the Krebs cycle, and it uses succinate as its substrate. But a structural analog of succinate, which is nothing but malonate, is capable of inhibiting this enzyme. What it does is it binds to the enzyme's active site, and it sort of has the same interactions with the enzyme active site as the substrate itself. This is possible because malonate has a structure that is similar to succinate. However, malonate cannot be catalyzed, which means there are no products formed. So it merely uses up the enzyme's active site. So it makes it seem like there are lesser number of enzymes than what is actually present. And in this way, it competes with the substrate for the enzyme and is known as a competitive inhibitor. Now let's see what happens to the enzyme activity in the presence of a competitive inhibitor. Imagine you add a fixed quantity of the inhibitor into the reaction mixture. The enzyme will now behave like it has a lower affinity for the substrate. And this is because there is a substance that is competing for the enzyme's active site. So the graph would look more like this. As you can see, the 10% velocity, which is reached at 2 millimolar substrate concentration, is now reached only at 3 millimolar substrate concentration. Similarly, the 20% velocity is reached only at 4 millimolar substrate concentration. So the apparent Km value has now increased. However, as the substrate concentration increases immensely, the maximum velocity that can be attained will not change. So the Vmax value in the presence and absence of the inhibitor is the same. However, the substrate concentration that was required to reach this Vmax value has now increased. We can understand this too by going back to our analogy. So we have the tennis ball with a red spot on it. And imagine you have like 5 army ants crawling on it. To this you add another 5 black ants. Now the probability of the red spot being occupied by the army ant or the black ant is the same. Similarly, when you have almost equal concentration of the substrate and the inhibitor, then the enzyme active site may be occupied by the substrate or the inhibitor. However, if you have the tennis ball swarming with army ants and you have 5 black ants on it too, then the red spot is most likely going to be occupied by the army ant, and the presence of the black ants will not matter anymore. Similarly, when the substrate concentration is very, very high, the presence of the competitive inhibitor will not matter anymore. And the enzyme's active site is most likely going to be occupied by the substrate molecule, and the reaction will proceed at the maximum velocity. Okay, so that's all about competitive inhibitors. But there's another type of inhibitor, a non-competitive inhibitor. These substances do not have a structural similarity with the substrate. And for that reason, they do not bind to the active site, but they bind to a different site on the enzyme. And they change the conformation of the enzyme, inactivating it, disabling it from being able to interact with the substrate. So how this inhibition works and how it affects enzyme activity is a bit different from that of a competitive inhibitor. But we shall not go into that right now. Okay, what else affects enzyme activity? Every enzyme is designed specifically suited for its function. It has a structure that is essential for it. So anything that destroys the integrity of the structure will affect the activity of the enzyme. So what determines the structure of an enzyme? The amino acids present in it. Based on the type of amino acids and how they interact with each other, the protein will fold. And this will form the final structure of the enzyme. So there are many tertiary interactions that stabilize the structure, such as ionic bonds, hydrogen bonds, hydrophobic bonds. So anything that will break or weaken these bonds will affect the activity of the enzyme. Okay, so what are the factors that break chemical bonds? Yes, heat. Heat can break the bonds that keep the enzyme structure intact. So if we plot temperature against enzyme activity, we get a bell curve, with the highest activity level being shown at optimum temperature. And as we move away from the optimum on either side, the activity will drop. So to a certain extent, enzyme activity increases along with temperature. And this is because heat enables enzyme-substrate interactions. But once it reaches the optimum level, any more increase in heat will break chemical bonds and denature the enzyme. And therefore, the activity level will drop, which is how we get the bell curve. Another factor that breaks chemical bonds is pH. pH, or the hydrogen ion concentration, can interfere with chemical bonds and can break them or even weaken them. So pH is another factor that affects enzyme activity. So if we plot pH against enzyme activity, you will get a bell curve that is quite similar to your temperature curve. And as you can see, the highest activity level will be at optimum pH. And as you move away from the optimum on either side, the activity will decrease. So that is all about factors that affect enzyme activity. Stay on to watch videos on how to solve neat questions that will appear from this chapter, the chapter Biomolecules.